Hi, am I? I know a lot of you have been confused about some of the differentiation between the different types of vaccines in 142, so this video is going to help clarify that. Also, I realized that the Project Lead the Way PowerPoint does not have a lot of detail, so I didn't really add much to it, but I added little indicators that might help a little bit. So if you want my version of the presentation, if you go into Canvas, and go into 1.4, 142. This PowerPoint that I'm going to go through in this video is there for you. But like I said, it just made it a little bit prettier with things that help me to understand it. So let's get into the presentation then. So you can also find this on my PLTW if you want. So it starts by telling us things that we kind of already know about vaccinations. We can get them subcutaneously, which goes below the skin, intramuscular, which is going into the muscle, or intradermal, which is going in between the layers of the skin. Oral vaccines are available, but only for a few diseases. You have an extra credit opportunity that will help you figure out why that might be the case, why it's not so popular, but we'll get to that later. So here are some characteristics of what vaccines should be. Obviously, we want them to have low levels of side effects or toxicity because we don't want adverse reactions. They do need to be effective in actually protecting us against exposure to a certain type of pathogen. It should stimulate a specific immune response with antibodies and or B cells and T cells. We want it to be long-term because it sucks to have to go back and keep getting booster shots, even though that is the thing that happens. So the longer a vaccine works, the better. It says should not require numerous doses or boosters. Obviously, there are still vaccines like that, but it's preferable to not have that. And obviously, we also have to think about the economics of it, right? How do we get vaccines to people? How do we take care of them and have a decent shelf life, but also making sure that that's not too expensive. It's also important that they're easy to administer so you can easily train people to give them. So here are the different types of vaccines. I find it confusing that they automatically put killed and inactivated in the same category. I get it though, because they kind of essentially do the same thing. Killed whole cells or inactivated viruses. Inactivated is like alive but can't reproduce or do anything. They're harmless, but they have specific antigens on the surface of their capsule, the viral capsule that protects them. It's kind of just like we have antigens on our red blood cells that say, hey, this is human, this is type A or B or whatever. Viruses have the same thing. So because the microbe can't actually multiply, you have to get the vaccine in larger doses because it can't reproduce and keep eliciting that immune response. And you're probably going to need boosters as well. So then one term that might be new to you is attenuated, which really just means weakened. So in that situation, we have a virus that we have weakened in some way. So it's not as good at performing certain functions. It is still alive, but it's weakened. So in other words, it's easier for our immune system to kill it off, but it can spread. But that can be a bad thing because the disease agent could go back and end up becoming pathogenic because we've just weakened it. So pros and cons with that. I use this little face because it's like it still functions, but it's not super sure about it. All right, so then these are the more small parts of vaccines. We can have them be toxoid vaccines, which is essentially using the toxin produced from some type of pathogen or by an antigen itself that elicits an immune response. So rather than using the entire organism, we're using a part. And that's what all of the vaccines on this slide are. They're using parts of a virus, not necessarily the whole thing. So in the first case, we're using toxins that is produced by the pathogen. But we can also genetically engineer certain organisms or certain antigens to elicit an immune response. So in this case, we're probably using a vector, which you might have heard of vector like in math or something before. A vector is just something that is used to do something else. So in this case, we're using a vector to transfer some sort of pathogenic agent. Oftentimes, that's going to be a plasmid. So we might engineer a plasmid, for example, and have it go into the organism as a plasmid versus just injecting these specific, protein or DNA that we're wanting from the pathogen, we've isolated a certain section and are transmitting it through the plasmid instead, so it's indirect. So that can be a lot of different types of vaccines. If you go to Google and type genetically engineered vaccines, you're going to find a lot of stuff because there are a lot of different applications. But just be thinking about things like we could use restriction enzymes maybe to build a certain genetic sequence that we could use to elicit an immune response. That's essentially what's happening with genetically engineered microbes and antigens. And then lastly, we have DNA vaccines. And in that case, the DNA itself is going to infect a recipient cells. So be thinking of like, we've got a host cell that's receiving foreign DNA that gets into the cell and starts producing proteins that can then elicit an immune response. mRNA vaccines kind of fall in that DNA category. But just remember that mRNA is one step removed from DNA. So in humans, we've got this native DNA. And then in order to get to RNA, we have to split the DNA and cut out the stuff that we don't want. So mRNA isn't the entire genome. So just keep that in your mind because I know you've been researching the COVID stuff as you go through. All right, so then the last thing is pretty self-explanatory. I'm sure that you've heard it. It's herd immunity. This just states that more individuals that are immune to a certain pathogen, that it will decrease the incidence of the disease and the occurrence of the pathogen. I feel like this diagram does a great job of showing. In the first, we have one person who's infected and nobody is immune to it in the population. So obviously that one person that has it can spread it very quickly to a bunch of other people because no one is immune. So there's no stopping point for the infection to spread. But if we have a bunch of vaccinated or immune individuals in a population, then it's less likely for that disease to spread. Now you can see in this diagram, not everyone is vaccinated. That's like the real world, right? Not everybody's vaccinated. But if the majority of people are vaccinated, it makes it less likely for the infection to spread. It's easier to stop the pathogen in that case, which is why we talk about vaccinations protecting the people that are immunosuppressed as well. Like maybe these people in blue are immunosuppressed or have some sort of allergic reaction to vaccines. These people in blue are the herd that protect the blue people from getting the pathogen, whatever it is. So I hope that that helps clear some things up for you. Don't freak out too much about the examples because there are a lot of different examples of vaccinations. As long as you can understand, are we using the whole virus or part of the virus, and is it alive or dead? That's what I care about the most. So I hope this helps.